Strokes get their name because of the rate at which symptoms come on. It's as if you're struck by lightning. Strokes are going to be caused by the disruption of blood flow, either depriving the brain of enough blood or causing blood to leak out and directly damage nervous tissue. In the first part of this lecture, we're going to talk about the difference between ischemic and hemorrhagic strokes, some of their similarities and some of their differences, and the way that they go about killing off brain tissue. In the second part, we're going to review the clinical symptoms that arise depending on location. For the most part, this is going to be a review of Lecture 2, so we'll kind of breeze through that. So when we're talking about strokes, there are two types. Either those that are caused by a blockage of blood flow, those would be ischemic, or those that are caused by the rupture of blood vessels, or hemorrhagic. The ischemic strokes are going to be the most common. As we can see in this plot here, we have 6 different sets of 3 bars. The different colors are just showing us different times, and the different sets are showing us whites and blacks, comparing ischemic and then hemorrhagic, either within the cerebrum or in the subarachnoid space. And as you can see here, there are some slight differences between the races, but there are marked differences between ischemic and hemorrhagic strokes. In ischemic strokes, we have the blockage of blood flow. And this can be for a couple of reasons. There could be a slow buildup over time that then leads to the blockage of blood flow. So this is our blood vessel here. So we could have this thrombus building up there, or we could have particles floating around, some sort of embolus that encounters a narrowing of the blood vessel and sticks in place. Either way, when there's impaired blood flow in that area where we're no longer receiving adequate blood supply, we're going to see an area of marked cell death. That's going to be our infarct. And then surrounding that, there's going to be an area called the penumbra, where we have some degree of cell death, depending on how long blood flow has been disrupted. With hemorrhagic strokes, what we see is a rupturing of the blood vessels and the leakage of blood into brain tissue. This is usually going to be more damaging than just a disruption. And even if you don't have a blockage of blood flow, at the site where you have herniation, usually at some branch point, all the downstream blood flow is lost as well. So even with a hemorrhagic stroke, you'll still get some degree of ischemia as well, because the blood that leaks out isn't going to be circulated to the rest of the tissue. Now here we can see that the greatest risk factor here is really going to be age. As with most things that we talk about in this class, the older you get, the more likely these are going to be to happen. So here we can see different age groups comparing men and women. Those are the different colors. And you can see clearly over there on the right, in the oldest age group, we have the highest risk of stroke. About 85% of strokes are going to be ischemic, and the other 15% will be hemorrhagic. In both cases, rapid onset, hence the name. Now, there can be just the buildup of that thrombus there to block blood flow. And in that case, you might get a gradual onset of symptoms. With hemorrhagic stroke, a severe headache is going to accompany the neurological symptoms that arise. And that's because the blood is going to cause just sort of non-specific stimulation of the dura, most likely. So most commonly we're going to see subarachnoid hemorrhages, and those are going to cause severe headache. But just the buildup of blood as it leaks out can increase intracranial pressure and cause headache. Diagnosis relies really on the sudden onset of symptoms, and then we use imaging to distinguish between ischemic and hemorrhagic, because CT is very good at picking up the blood that flows out, and it's very fast and cheap. That's the first set of imaging that's going to be done. You don't want to wait for an MRI. So CT is first used to decide do we have blockage or breakage of the blood vessels. If we have blockage, well, then we're going to treat with anticoagulants. We're going to use anticoagulants to help break up this cluster. But if we were to apply anticoagulants when we have hemorrhage, you would make things worse. So we use CT to determine whether there's a bleed or not, and we'll follow up with MRI. The underlying causes are pretty similar. Hypertension can cause either one of these. As you have atherosclerosis, that can of course increase the likelihood that you have blockage of blood vessels, and there are some conditions that make it more likely for blood vessels to rupture, like cerebral amyloid angiopathy. We'll get to that in just a little bit. The most common site where you'll find ischemic strokes will be along the distribution of the middle cerebral artery, just like for our secondary brain tumors. Because the middle cerebral artery is continuous with the internal carotid, any little bits that are floating around out there in the periphery are going to enter the brain through the internal carotid and then continue along the middle cerebral artery more commonly than branching off to hit the anterior cerebral artery. The prognosis really, of course, depends. So comparing the two types, ischemic strokes tend to have better prognosis than hemorrhagic, and in both cases the loss of consciousness is indicative of poor outcomes because one of two things have happened. You either have far more widespread damage to the cortex that creates that loss of consciousness, or you have damage in the brainstem leading to the loss of consciousness. Now, of course, because we have those vital functions carried out in the brainstem, damaging the brainstem naturally produces a poor prognosis. Now, the whole reason that blockage of blood flow is so devastating to neurons is because they're greedy. The brain uses a lot of energy. Although it accounts for about 2% of our body mass, it's going to use about 20% of our oxygen and about 25% of our glucose. So by weight, this is a very greedy organ. Now it needs a lot of glucose because that's the principal energy source for nerve cells. Of course, we're going to be using glucose and oxygen to create carbon dioxide, water, and 30 plus or minus a handful of ATP. This is exactly like what you learned in your general biology classes, good old cellular respiration. We'll get some glycolysis in the cytoplasm, and then we'll have our citric acid cycle and oxidative phosphorylation within the mitochondria. And in the end, we get a whole bunch of ATP. Now, we think that glucose is the principal energy source for the brain because of the respiratory quotient, or the ratio of oxygen consumed divided by carbon dioxide produced. If you're using glucose as 100% of your fuel source, you should be making just as much carbon dioxide as you are consuming oxygen. So your ratio should be 1. And when you look on the left side of that plot there, that's for brain tissue, and you can see it's pretty darn close to 1. Moving into more peripheral tissues like arm, leg muscle, the respiratory quotient drops, indicating that there are other sources of ATP production, such as ketone bodies, lipids, for example. Now, when we're talking about energy usage in the brain, this is the protein that's using the lion's share of ATP. This is the sodium potassium ATPase. What it's going to do is bind 3 sodiums that are floating around inside the cell. It'll bind a molecule of ATP, break it, so it's going to hydrolyze ATP. That free energy that's released is going to drive a change in the structure, causing the release of those 3 sodiums. 2 extracellular potassium will bind. That binding event causes another change in conformation that spits off that phosphate, reorganizes the protein, and causes the potassium to fall off inside the cell. So what we're doing is moving a net 3 sodium out and 2 potassium in for every 1 ATP that we spend. The reason we have to do this is because the activity of neurons relies on the passive movement of sodium into the cell and potassium out. So since we're always letting sodium come in and letting potassium leave, we have to reset our ionic imbalance. And we'll talk a whole lot more about this next semester. But all you need to remember, there's a lot of sodium outside, a lot of potassium inside, and we want to keep it that way so that neurons continue to function. So we're going to spend about half of our ATP just on this protein. Now the amount of ATP that a neuron spends depends on how active it is. Every time it fires that action potential, it takes about 3 to 4 milliseconds, and it's going to burn just about as much ATP as we do for 1 second of rest. In other words, we're spending about 250 times more ATP whenever we're active. So active neurons use a whole lot of ATP, and thus neuron activity is going to need a whole lot of ATP. So when we have disruption of blood supply, we run into insufficient ATP. This is going to kill neurons in several ways. So when we run out of our ATP, one of the things that we don't do is fuel that sodium-potassium pump. We need ATP to drive our sodium-potassium pump. In the case of stroke, no more ATP production because we don't have blood bringing the oxygen and glucose that we need. We don't run our sodium-potassium pump, so we don't maintain normal resting membrane potential. The neuron is going to depolarize, and that's going to cause it to be more active, burn more ATP, and create more of a problem. That increase in activity is going to do a couple of things. It's going to spit out a whole lot more neurotransmitter. Now, one of the more common neurotransmitters would be glutamate. Glutamate is an excitatory neurotransmitter, so that is going to drive more activity. And as these neurons are depolarized, what that does is increase the amount of calcium within the cell, which of course drives the release of neurotransmitter and drives further activity. You can see this is a vicious cycle. So as we run out of ATP, the cells just depolarize, spit out a bunch of neurotransmitter that causes further depolarization, and then we have this dangerous increase in calcium. Calcium levels are tightly regulated and kept extremely low in cells. Part of this has to do with the amount of phosphate floating around, and another part of it has to do with the fact that calcium is toxic, at least at high levels. So if we have a whole bunch of ions accumulating within a cell, water is naturally going to follow. It's osmosis. So as water flows into the cell, that cell is going to balloon up until it pops. So you can get osmotic necrosis. It can swell up so much that the cell membrane ruptures. Alternatively, if the cell doesn't rupture, that increase in calcium can permeabilize our mitochondria. Now the mitochondria do a lot of really wonderful things for us, and they are necessary for cells to survive. So when we have damage to our mitochondria, the cell detects this as a severe enough issue to undergo apoptosis. So whenever we permeabilize our mitochondria, that triggers cell death. So if we run out of ATP and we don't fuel our pump, we depolarize. That depolarization drives further depolarization because it spits out more glutamate, and glutamate depolarizes the neuron. That depolarization leads to an accumulation of calcium. That depolarization opens up calcium channels, and we can't fuel our calcium pumps because they run on ATP as well. So calcium builds up, and when calcium builds up, the mitochondria try to clean it up. So they create little holes. They permeabilize to clean up the calcium, and when they do that, toxic stuff leaks out and the cell dies. So it could be through swelling, it could be through damage to the mitochondria. Either way, it ends pretty much the same. Neurons are lost. Now all this damage that's occurring is going to be detected by glia, and we're going to have reactive gliosis, just like in Lecture 1. When astrocytes become reactive, They stop buffering potassium, so we get an increase in extracellular potassium. Just like in previous lectures, that is going to depolarize the neuron as well. You can see contributing to this vicious cycle. Astrocytes also decrease the amount of glutamate that they are taking back up. So the glutamate that we spit out is going to remain at the synapse for longer, further driving activity in the neurons and thus contributing to the toxicity. So it all kind of works together. When neurons don't have enough blood, oddly enough, they become more active. And that activity leads to excitotoxicity, and that's exactly what we just went through here. Now, not every blood vessel blockage is going to be complete. Some of them are going to be very minor, and they can be caused by a spasm in the blood vessel, or just a brief blockage that dissolves. And these can create what we call a transient ischemic attack. So if we think about, let's call this blood flow, and there's 100% over time, some arbitrary units of time with a stroke, we get an immediate and prolonged decrease in blood flow. But with a transient ischemic attack, we get a very brief blockage of blood flow. So we get transient symptoms. And although they go away, they should be taken as a warning sign. Within about 3 months, 17% of patients with a transient ischemic attack are going to have a stroke, full-blown stroke. And after 4 years, about a quarter of those with a transient ischemic attack will be dead of a heart attack. Now, all that damage that occurs as a result of stroke is, of course, going to trigger an inflammatory response. When we have neurons dying off, here's a dead neuron, so this one's alive and well, and this one not so much. We got to clean up this damage. So, of course, we're going to, that's a damp there, so we'll have some damage-associated molecular patterns, just like we saw in Lecture 3. They're going to trigger our microglia to clean up the mess. They then become reactive and consume the dead cells. This inflammatory response, just like in Lecture 3, is going to cause a little bit of swelling, could cause brain herniation if it's pronounced enough. So that can contribute to the damage. So we can have our primary damage from the stroke and the disruption of blood flow, and there's also secondary damage that occurs after the fact, part of the cleanup. Now, it's very critical to treat any disruption of blood flow as quickly as possible. The faster you get to it, the more nervous tissue you can spare, so recognizing early warning signs are important. And they're varied, as you would imagine. It all depends on the location. But a sudden onset of neurological signs should be taken as a warning sign of stroke, especially if there's a history of hypertension, for example. Now, the hemorrhagic strokes have the additional issue of blood leakage. They'll also get some ischemia as well, but at the site of hemorrhage, we have blood leaking out. Blood is toxic to neurons, like many other things. So at branch points, where we have typically a weakening of the blood vessels, rupture there causes blood to leak out. Now, blood isn't supposed to be in brain tissue, so this gets, of course, degraded by our microglia, and it's going to form heme. There will be some iron floating around, and what these are going to do is damage neurons by creating free radicals. Peroxides, for example. What free radicals are molecules that have an unpaired electron. And so what they do is get rid of that electron. So they go and break apart the covalent bonds that are holding together macromolecules of the cell, like lipids, proteins, DNA, for example. So they're going to non-specifically damage neurons. Now, the reason that we have breakage here could be because of high blood pressure. So that hypertension is going to affect the vascular smooth muscle cells. So rather than remaining as smooth muscle, whenever there's increased pressure on them, they respond and they differentiate into a different type of cell. Some of them are going to become little collagen factories. So they'll make a whole bunch of collagen to try to toughen up the blood vessel. Because they're losing their muscle and creating a whole bunch of collagen, the blood vessels become very stiff and they're more prone to rupture as a result. Others can adopt more of a macrophage phenotype and stimulate an inflammatory response. This can lead to necrosis within the blood vessels called fibrinoid necrosis. Some folks are also going to have the issue of increased A-beta production or impaired clearance of A-beta. A-beta is just amyloid beta. Amyloid just means starch. This is a protein that forms these big starchy balls called amyloid in Alzheimer's disease. Now when you see big balls of amyloid in the brain, that's indicative of Alzheimer's disease, except when it's not. When you see them in the blood vessels, that's indicative of cerebral amyloid angiopathy. So the buildup of this A-beta also stimulates an inflammatory response, leading to the weakening of blood vessels, making them more likely to pop. When blood leaks out, that then damages neurons through heme toxicity, where we create these free radicals, and free radicals just tear apart the cell. Now the good news is that's pretty much all the new information for this lecture, because the clinical syndromes are going to be based on all that stuff that we talked about in Lecture 2. It's all about the location. Most commonly, you're going to have the middle cerebral artery being affected here. And there's a few different divisions of it. There's the superior divisions that are going to serve the frontal, parietal lobes, the inferior divisions that are going to hit your parietal and temporal lobes, and there's some deep divisions that are going to serve the basal ganglia and the internal capsule there. Now depending on which part we block, that's going to determine which signs do we see. So if you're blocking the frontal lobes, you might expect some motor or behavioral abnormalities, and over there in the parietal lobes, maybe more somatosensory abnormalities, or even perhaps some visual abnormalities as well. So we've seen this before. This is our clock drawing task. This is an example of hemi-neglect. You can watch this in the slides all on your own. On the other hand, we have Broca's aphasia here. So there's a couple different language areas. There's still Broca's area, Wernicke's area. They're the same. This is the same video. You've seen these. Broca's aphasia, Wernicke's aphasia. The anterior cerebral artery is going to serve a little more medial than the middle cerebral artery. Despite its name, the middle cerebral artery is going to serve more of the lateral surface. So in this image, on the bottom, you can see the divisions of the middle, anterior, and posterior cerebral arteries. The middle cerebral artery is colored in yellow. You can see that's hitting the lateral surfaces in all of our lobes. The more extreme ends are going to be served by the anterior and posterior cerebral arteries, and the medial aspects are going to be hit by those as well. The anterior cerebral artery is colored in blue, and the posterior in red. Now I want you to pay attention to that bottom right illustration there. There's a fiber band called the Corpus Callosum, which is served by the anterior cerebral artery. Whenever there's occlusion of the anterior cerebral artery, then the ability for the two hemispheres to communicate with one another can become perturbed because of damage to the corpus callosum. All the corpus callosum is a big white matter tract that's going to connect the two sides of our brain. So here's the two hemispheres. You got your left, you got your right. They're a little wrinkly, something like that. But there's a fiber bundle running underneath there that connects all of our cerebral cortex together, left and right, so the two sides can function together. When you have damage to the corpus callosum, you can have a condition that we call alien hand syndrome, where the two hands don't work together. They can sometimes oppose each other. So here he's trying to solve a puzzle. And he's trying to solve a puzzle. He can use one hand or the other, and you'll see they're disagreeing on where that piece should go. His hands are kind of fighting each other. Now the left side of our brain is usually more dedicated to language, and so the right hand, which is controlled by that, might not have as good of an idea about spatial relationships, because spatial relationships tend to be more encoded on the right side. So the left hand has a better idea of how to solve these puzzles. This is kind of a long video, so you can follow up. But basically, his left hand's going to do a better job. In the posterior cerebral artery, we're going to be serving more the back of the brain. So the name makes sense. Anterior more the front, posterior more the back. So here we're going to see some. It could be difficulties with vision. It could be difficulties purely with reading, or if we have problems with the deep division, we can get issues with the thalamic function, or damage to the midbrain, creating that third nerve palsy that we've seen before, and any problems with the posterior circulation that serves the brainstem, which of course includes the midbrain, pons, medulla, and cerebellum there. They can create their own host of neurological deficits. Because we have our long tracts running through there, or the tracts that go from the head to the rest of the body, like your corticospinal tract, also called the parental tract, your posterior column medial meniscus, and your spinal thalamic tract. All of those are running through the brainstem. That's how they get from body to cortex and back. So infarcts in the brainstem can also damage these tracts, creating either motor deficits, sensory deficits, or some combination. Now, of course, just like in Lecture 2, the midbrain, pons, medulla, they're going to have different functions, and damage in different areas will produce different deficits. Damage in the midbrain could lead to coma, could lead to cranial nerve palsies that control the eyes. Damage to the cerebellum will tend to create ataxia. So here we're going to see damage to the left cerebellar hemisphere. So you can see his right side is moving just fine. This task is fairly easy to do when you have an intact cerebellum. Now I want you to notice the intention tremor. So as he's trying to hold his finger still, that becomes difficult for him to do. And then finding his way to the nose becomes difficult as well. Now the cerebellum controls the ipsilateral side of the body, so we can tell this is his left cerebellum. And so that very easy automatic movement that we can all do becomes difficult for this gentleman because of damage to his cerebellum. Damage in your midbrain, or even potentially in the pons, can lead to coma because that ascending arousal system is lost. Damage in the more anterior portion of the pons that affects your corticospinal tract can create that locked-in syndrome. And of course down there in the medulla, where we control respiration and cardiovascular function, can lead to death. We've got a few review questions here. Have a look at those before class. But what we're going to do is review the mechanisms of cell death and kind of distinguish ischemia and hemorrhage. But if you've got basically this and the last board on there, you're probably good to go. If you have any questions, send me an email. I'll see you in class.